All right, class, remember last time we were talking about crossing between two different types of organisms, genetics, right? So remember that, guys? Remember? It's fun times. Okay, great. So let's just remind ourselves really the steps in this process of using our Punnett square, right? So we're talking about guinea pigs, right? So in the guinea pigs, we got short hair, which has an S there for it, and that one is also dominant. So how do we know that something is dominant if I'm reading a genotype?
It's a capital letter.
Capital letter, good. Okay, remember that? Okay, great. Okay, so got capital letter for dominant long hair, and we're going to complete a cross where one guinea pig is big S, little s. What does that mean? What does that mean about this guinea pig? Big S, little s. Who remembers? It's got one big S and one little s, right? So if one big S is long hair,
dominant and one
recessive, right? Exactly. One's dominant, one's recessive. Good. So if I have a dominant and a recessive, right, the dominant takes over. Remember that from last time. Great. Awesome. So we're crossing that one with one that is little s, right? So little s means they're both recessive, right? So what does that look like? If it's not long hair, it's
short hair.
Exactly. So we have a short hair and a long hair, and we're going to cross these two. Okay, so let's look back at our steps from last time. We already have these written out for you, so you're ready to go. All right, so we're going to write the parent alleles on the top two. So what would I put here? Big S, right? And then this one will be what?
Little s.
Little s, right? Because I'm looking at this first one, right? This first guinea pig. On the other side, I'm crossing it with the other one, right? So on the other side will be what?
Little s, little s.
Good, little s, right?
Mr. Mamara, should we be writing this down?
Yeah, you can. You can. Go ahead and jot that down if you're confused. I think that's a good idea. Sure. So we're going to take the big S and the little s. We're going to cross that, right? And then I'm going to do the same thing here. And I'm going to do the same thing for all of these, okay? That's the same process, okay? You good? All right. We're going to get working.
Okay guys, so we've just finished our notes on Punnett squares and all of our vocab. So I want to make sure that we understand how to use a Punnett square. So really quick, I just want you to slide your notes under your notebook, put your pencils down, because I'm going to model this for you before we do an example together. I want you to see the thought process through this. So I'm going to read my question first. It says, in guinea pigs, short hair, which is capital S, is dominant to long hair, which is lowercase s. Complete a cross where one guinea pig is capital lowercase and one is two lowercase to calculate the genotypic and phenotypic ratios. So that's a lot of words. These questions are going to have a lot of words. So I know that I need to break these things down first. So when I look at this, I'm going to underline what I'm given, and I'm going to circle what I'm looking for so that I don't get lost in all the extra stuff. So I'm reading through here. Okay, well, it's important to know that S is dominant, capital S is dominant for short hair. Long hair is lowercase s, so I'm going to just make a note for myself here that that means recessive. Okay. I have a cross where one guinea pig is big S, little s. I need to remember that that is heterozygous, because I have a dominant and a recessive. But then my other parent is little s, so that's a homozygous recessive because they're lowercase, so that's recessive, and they're both lowercase. Then I need to circle what I'm looking for so I don't get lost, and I'm really paying attention to calculating those genotypic and phenotypic ratios. But I can't do that until I set up my Punnett square, right? That's the whole reason that we're using those. So when we're looking at a Punnett square, we have a couple steps to follow, and I know that my first step here is to write the parent alleles on top and left side. They will always go on the top and left side, so the more I get used to that, the easier it's going to be. So again, I had one parent that was heterozygous, so I'm going to take that allele, or that, that genotype, and I'm going to split it up one allele per box on my Punnett square. So I'm going to put my capital S first, and I'm gonna put my lowercase s over the second box. Okay, so I've taken care of parent 1. So then my other one was lowercase. Lowercase, I'm gonna do the same thing down the side where this is parent 2. So once I've done step 1, I've written in the parents, then I need to remember that each box will inherit the allele from the column and row that it's on. So when I look at this, I'm going to say, okay, well, I've got to make a genotype in each of these boxes. So I'm going to say, okay, well, we've got on my column, capital S, from my row, a lowercase s. I need to remember that dominant always comes first. So since I do have a capital here, I'm going to write that one first. Okay, great. So then I'm going to do the same process here with the second box. Well, this is a lowercase and a lowercase. Doesn't matter which one comes first. And then I'm going to repeat this process for the last 2 boxes. Okay, so now that I've filled out my boxes, I can then go into these genotype and phenotype, these ratios that we're looking at. I need to remember, deep down, genotype, we are looking at the gene or the DNA. So this is looking at those 2-letter pairings that we've been studying through this lesson. So I'm going to say, okay, well, I need to look at each box, and I'm going to write down the genotype in those boxes. So I've got a capital S, lowercase s. I've got a lowercase, lowercase. Well, I've got another capital, lowercase, so I'm going to put a 2 there. I've actually got a 2 for my lowercase, lowercase. Yes, Jordan?
I have a question about the parent 1 and parent 2. Do you have to be in that same order? Like, can I put them opposite ways? Or?
So let me really quick finish this example, and I'll come back to your question because we're going to do a we do, and we'll walk through that as well.
Okay.
It's a good question, though, so hold on to it. So we've got our genotypic ratios here, but then remember for phenotypic